our guests and all participants. My name is Muchane, a research scientist at the National Museums of Kenya. I am here to give a talk on the role of citizen scientists in mushroom research and conservation. Welcome. Before we look at the role of citizen scientists in mushroom research and conservation, we will first see what is citizen science. Citizen science is scientific research conducted in whole or conducted in part by non-professional scientists. These non-professional scientists may even include the amateurs. In other words, we can define citizen science as an active involvement of general public in correcting or active involvement of general public in correcting and analyzing of scientific data. These non-professional scientists are usually volunteers with a passion in an area of study. They can be local conservationists, they can be tour guide, they can be scientists, they can be students, farmers, or any other person interested in contributing to science. These non-professional scientists are the ones we are calling citizen scientists. Why should we use citizen science in research and conservation? First reason why we should use citizen science in research and conservation is that it is a very cheap way of acquiring scientific data. Citizen science program utilizes people on the ground. It thus reduces the cost of accommodation. It also reduces the cost of transportation, making it a very cheap way of acquiring scientific data. The second reason why we should utilize citizen science program in research is because one can cover large area within a very short time. When using citizen science program, one is able to recruit a wide range of people from different locations, making it a very fast way of acquiring scientific data. Citizen science program also enables scientists create local awareness on the role of science in development. For us in biodiversity research and conservation, for example, when we use citizen science program, we are able to create awareness on the role of biodiversity in our ecosystem. We are also able to create awareness on why the local community should support biodiversity conservation programs. When we use the citizen science program, we are also able to enlighten the citizen scientists, or we are able to enlighten the local communities, and in doing this, we are able to improve their scientific literacy. We are also able to improve their capacity, and now these people can contribute to science. They can also contribute to conservation significantly. When we have now enlightened local community, the adoption rates of scientific results as well as conservation technology is increased drastically. This is because we do not have now to convince these people to know the importance of a given scientific result, because they are already aware of the importance of this result in their ecosystem. They are also aware of the consequences if these results are not implemented. Although citizen science programs have been used widely in research and mapping of various taxa like birds, mammals, plants, the programs are rarely used in fungi research. That's why we came in early 2019. We initiated a Mushroom of Kenya project with the aim of understanding the role of citizen science in mushroom research and conservation. This work was motivated by lack of macrofungi diversity data. It was also motivated by lack of macrofungi ecological data in our Kenyan ecosystem. And due to lack of this data, there has been no effective management and conservation strategies that have been developed for these species. The consequences of this is that the fungi has remained overlooked in major conservation initiatives, and yet we know that the macrofungi diversity or the mushroom diversity, it is very key in sustainability of our ecosystems. Before we continue, I want to give a brief description of what macrofungi are in general. Fungi are living organisms. They are not plants. They are not animals. Fungi are classified in their own kingdom, the kingdom fungi. In this kingdom, we have two categories of fungi. We have the macrofungi. These are fungi that we cannot see with our naked eyes. We can only see them clearly using a microscope. The second category of fungi is called the macrofungi. These are fungi with a visible fruiting body, or they are fungi we can see with the naked eyes. There are different categories of macrofungi. We have the mushrooms, we have the puffball, we have the bracket fungi. However, most of us know the mushrooms. Mushrooms are usually characterized by having a cap. A cap is the top part of the mushroom. It can be smooth, it can be scaly, or it can have scales. It can be hairy. Beneath the cap, we have the spore-producing tissues. These can be gills or they can be pores. Beneath the gills, we have the stipe or the stem of the mushroom. The stipe supports the cap and connects the cap with the mycelia. Beneath the stipe, we have the mycelia. These are thread-like roots. They attaches the mushroom to the substrate or the media where the mushroom is feeding from. In general, a mushroom is usually made of a dense mass of mycelia, or what we are calling the thread-like roots. Why do we study mushrooms? We study mushrooms because they are key in sustainability of our ecosystem. In their natural condition, most of the mushrooms are decomposers. They release nutrients from the dead organic material back into the soils, and in doing this, they make nutrients available for the living plant community. As a result, they are able to maintain a stable plant community. Secondly, some mushroom species form mutualistic relationship with the plant. From this relationship, mushroom species help the plant take up nutrients and water from the soil. They also help in control of root-related diseases, and in doing this, they help maintain a productive and healthy plant community in our ecosystems. Another reason why we study mushroom is because they are key in climate change mitigation. The density hyphae of these mushrooms in the soil help in development of micro and macro aggregates. We know these aggregates are very key in the storage of carbon in our soils. We also study mushroom because they are very key in cleaning of our polluted environment. Mushrooms have ability to break down the chemical compounds found in major pollutants in our environment, and in doing this, they help maintain a clean environment. We also study mushroom because some of these mushrooms are parasitic to our plants. They are also parasitic to our crops, and they cause significant economic losses in our crop and tree production. So we have to study these mushrooms or these fungi so that we can be able to manage them and maintain productive ecosystem and productive agriculture. Mushrooms are also very key in improving local livelihood. Mushroom cultivation enterprises, for example, they provide both food and income to the farmers, and in doing so, the livelihood of these farmers is usually uplifted. After short descriptions of what mushrooms are and their importance, we are coming back again to see why we should engage citizen scientists in mushroom research and conservation. Mushroom is usually a very unique taxa. It is unique in such a way that it has a very short lifespan, a lifespan of 1 to 20 days. It is therefore very easy to miss a species if you come for correction or if you come for sampling when its lifespan has ended. Mushrooms are also unique in such a way that they are affected by season. Mushrooms mostly occur during the rainy season, and not all of them will appear at the same time during the rainy season. There are those which appear at the beginning of the season, there are those who appear at the end of the season, and there are those which will appear at the middle of the season. This means that the short survey usually carried by scientists will always underestimate the mushroom diversity in our ecosystems. That's why now we need to engage the citizen scientists. These are the people on the ground. They are the ones who can document these species when they are sprouting. They are the ones who can document these species across the season. And they are the ones who can now document these species before their lifespan has ended. We are also using citizen science program to enable us create local awareness on the importance of mushroom community in our ecosystem. And we know when we do this, we are going to improve the scientific literacy of our local communities, and we are going to enable them contribute significantly to mushroom research and their conservation. We are also using citizen science program to help us generate macrofungi diversity data, a data that can be used in development of management and conservation strategies for these species. We also want to gain data that can help in expanding the mushroom industry in Kenya. We also want to gain data that can be used in developing effective afforestation program, especially for tree species that form mutualistic relationship with some of these fungi species. At the same time, we want to generate data that can be used to develop a field guide that can be used by both the scientists and the citizen scientists in describing and identification of mushroom or macrofungi species in Kenya. As I said before, in January 2019, we began a pilot study to test the effectiveness of citizen science program and iNaturalist data collection tool in mushroom or macrofungi data collection. In the next lines, I will show the potential citizen science program and iNaturalist data collection tool have in mushroom or macrofungi data collection. In this pilot study is currently being done in Aberdare Forest. In this pilot study, we first developed a mushroom monitoring program. In this program, citizen scientists are collecting mushroom specimens from already established permanent plots. This monitoring program is being tested in 2 sites in Aberdare Forest. The first site is in southern Aberdare, that is Greater Forest. In this site, we have recruited 7 citizen scientists. This far, they have managed to collect over 3,000 mushroom specimens. These are translating to over 500 mushroom species. In the other site, which is in West Aberdare, the Greater Forest, we have also recruited 7 citizen scientists. This far, they have managed to collect over 1,000 mushroom specimens, and these specimens are translating to over 200 mushroom species. We also developed a Mushroom of Kenya project in the iNaturalist. For those who have not come across the iNaturalist, iNaturalist is a free online platform. It helps in recording and sharing of biodiversity observation using photograph. The use of this program or platform will be discussed later in details in this webinar. When we developed this iNaturalist Mushroom of Kenya project, we developed it to capture mushroom data collected all over Kenya. We wanted to capture all mushroom data collected by other passionate citizen scientists. This far, the project has received over 2,500 mushroom specimens. These are translating to over 400 mushroom species. From this data collected by citizen scientists, using iNaturalist data collection tool, we are now able to see how mushroom diversity is distributed across Kenya. From this map, we can see how mushrooms are distributed all over Kenya. We can see mushroom observation in all regions in Kenya, with the exception to northern eastern Kenya. When we translate this data into a graph, we can see very high mushroom density in central Kenya compared to other regions. This is understandable because this is where we have a detailed monitoring program in Aberdare Forest. Using this data collected now by the citizen scientists, we can now see how mushrooms are distributed across different counties in Kenya. In this slide, we show how mushrooms are distributed across the Kiambu County. Using this data still, we can also see how different macrofungi species are distributed across Kenya. From this map, we can see how Agarika species are distributed across Kenya. It is very interesting to see Agarika species even from the northern part of Kenya. From this work, we have discovered a wide range of macrofungi species. The species have ranged from mushroom, toadstools, puffball, stick-and horns, polypons, asters, and the like. We have also discovered a wide range of edible and medicinal species. Some of these species are being captured to preserve them for future research and for future use. From this work, it is clear that many of these species we discovered would have taken long before being discovered or would have taken long before being documented. We therefore thank the Citizen Science Program and also the citizen scientists for this great progress. In conclusion, I can say that Citizen Science Program and the iNaturalist data collection tools have potential in revealing macrofungi species in our Kenyan ecosystem. From the Mushroom of Kenya project under the iNaturalist, we have discovered over 400 mushroom species. These are from over 2,000 observations. I cannot confirm this is the highest number of macrofungi species ever discovered in our Kenyan ecosystem. From the detailed monitoring program in Aberdare Forest, we have discovered over 500 mushroom species from over 3,000 observations. I can also confirm that this is the highest number of macrofungi species documented in our Kenyan ecosystems. We are therefore calling each one of us to join this citizen science program. The data from your observation and my observation will be very key in developing management and conservation program or technologies for macrofungi species in Kenya. The data will also be very key in expanding mushroom industry in Kenya. This data we are collecting will also be very key in supporting a forestation program in Kenya, especially for the tree species that form mutualistic relationship with some of these mushroom species. This data will also help us develop a field guide that can help in descriptions and identification of mushroom from our Kenyan ecosystem. Through this program, we are also going to gain knowledge in mushroom taxonomy. This knowledge will help us contribute significantly to science. We can also get engaged in identification of mushroom species submitted to iNaturalist online repository. Let us therefore join hands in making our mushroom diversity known to enable their sustainable management for healthy and productive ecosystem in Kenya. As I conclude, I wish to thank the iNaturalist team and the California Academy of Science and the Natural Geographical Society for supporting this webinar and for supporting this work. I also wish to thank the staff of National Museum of Kenya, Nairobi University, and all Kenifo staff for supporting this work. I also wish to thank all citizen scientists who have contributed their time and resources to make sure mushroom research does not lag behind. Let us keep up the good work. I also wish to thank all of us for joining and listening. To all of us, thank you, and let us keep mushroom research ongoing.